So good morning to all.
Good morning, sir.
I hope we can start our session. Right now, I'll be handling the paper that is or the course molecular biology and RDNA technology, which is a third core course in the microbiology syllabus. And anyway, as the first new students, you might be interested in getting a copy of the complete syllabus of microbiology, M.Sc microbiology. It is already given in the department website, and I will be adding a link to the Moodle platform. Here in this paper, you will have only the concerned syllabus of the third core course, that is molecular biology. And I'll be adding another link to a PDF file which contains the entire syllabus of M.Sc microbiology under the CCSS scheme, which you can go through and you can see what are the papers that you are going to deal with during the next 4 semesters during the course duration. And as we discussed yesterday, in this new era of corona and online learning, you'll have to explore new methods, especially in the digital platforms for our learning and evaluation methods also. So anyway, we developed a Moodle-based platform, and I hope most of the faculty members will be joining this platform and they'll be using it. Maybe there'll be some problems with other faculty members who are not so familiar with this platform, but still we'll extend all the support and we will try to systematically use such learning platforms like Moodle for this coursework. So this is the first course that we are going to start in this semester, that is the molecular biology and RDNA technology. And I think all of you might have gone through basics of molecular biology in your UG curriculum. And naturally, the basics were already known to you. But at the PG level, let's see what are the contents of the syllabus. I think it may not be visible to you.
Yes, sir.
Good. So this is about the syllabus. In the first unit of the molecular biology, we will be discussing some general concepts about the genome organization, prokaryotic and eukaryotic genome organization, and some of the concepts like C-value paradox, pseudogenes, then gene families, then the structure of the DNA, Watson-Crick model of the DNA, and DNA replication, basics of DNA replication, and other things. So this unit is actually a basic unit which you might have already gone through in your degree syllabus or degree curriculum. So I think we don't have to spend much more time in explaining the things that you already learned, but we will add something, or we will supplement the existing knowledge that you have, that you already have, and then we will move forward to the next units, like the transcription. The second unit is on transcription and post-translation modifications and the different concepts related with that, including the operon concepts. And the next third unit is on translation and post-translation modifications. Then fourth unit is on oncogenes and cancer, then the related aspects or concepts. And the fifth unit will be handling with PCR. Then the next unit is on restriction endonucleases and or genetic engineering, RDNA technology or recombinant DNA technology. And finally, there will be a seventh unit that is on pharmaceutical products of DNA technology and related concepts. So this is the syllabus that you are going to deal with during this semester. Now, actually, is there any problem with the students in joining? Because there should be 13 students. I think only 9 joined. Nobody contacted me yet. Anyway, let's start. So in the first unit, uh, this genome organization, it is already known to you, that the histone proteins and other things, it's already known to you. We are not going into the that much details or from the basics, because it's already learned in your degree syllabus. Right now, I think we will start with the C-value paradox. Maybe it is not mentioned in your degree curriculum. Is it there?
No,
Did
sir.
you
No, sir.
already? No. So we will learn the concepts which is new to you, and the other ones maybe we will skip the already learned concepts will be skipped, and the remaining portion, that is a anything new that needs to be supplemented, that will be taken during the lecture sessions. And another one, if you are interested in taking a portion as a seminar topic, that we will decide this week itself. And for that purpose, we have provided a link here. Now, I will visualize it to you. Okay. Now onwards, this link will be available to you. So this is a Google Doc sheet or Excel sheet on the cloud. All of your names will be here. You can type in the concerned topic that you are going to handle for the seminar and assignment in this coursework. And you have to enter the name one by one. So everyone need to access this link for submitting your seminar topics. After going through the syllabus and select an interesting topic on assignment submission and seminar presentation. So it will be the same topic. So whatever topic you are selecting, you will prepare an assignment on that one. And after submitting the assignment, you will schedule a date for the seminar, online seminar presentation. And on the same date, on the same topic, you will present the seminar. So that is that is how the system is going to work. So right now, within this week itself, by before Friday, you are going to access this link and then fill in this form with your seminar topics. And based on that seminar topics, your subsequent sessions will be arranged. And all the topics that is not actually included in the seminar topics, and if it is that needs to be addressed in the lecture sessions, that will be handled during the lecture. Okay. So this is about the seminar topics, and now we will come to the first topic that we already selected, that is the C-value paradox. In the case of C-value paradox, let's explain what is C-value paradox. The term C-value that refers to the amount of DNA contained within a haploid nucleus, that is, in a gamete or one half of the amount of the diploid somatic cell. Usually, you know that there are in eukaryotic cells, usually if it is a diploid cell, it is having two sets of the same genome content. So in a haploid cell or a gamete, we have only one set of the concerned chromosomes or one set of the genes. So C-value actually refers to the one set of the genes. So in case of humans, for example, we will say that there are 23 pairs of the chromosomes, and it is not the 46 or the entire content of the genetic material, but the C-value actually refers to the entire content of the 23 pair of the so 23 chromosomes, not the pair. So a single set of the concerned for the haploid number of the DNA content. That refers that is actually referred by the, or which is meant by the term C value. Okay, so that's C value. In some cases, notably among the haploid organisms, the term C value and the genome size are usually used interchangeably, because in case of diploid it is half, in case of haploid cell or a gamete it is the same. The C value is the same as that of the total DNA content. So the C value actually refers to various terms, like many authors or many scientists were offering differently, like characteristic value or constant value or content value, complement value. Various terms were actually assigned to the C. So anyway, C value is actually a characteristic or constant complement value in case of a value that is actually a characteristic of or a constant value that is used for referring to a genome content. The letter C stood for constant, that is the amount of the DNA that has been characteristic of a particular genotype. So finally we can conclude that it is a constant value. C is actually it's a constant value that is applicable for a particular cell type. So it may not change. So this is a graph that actually shows the genome content or the genome size of the various organisms. Okay. So here you can see on the top we have mycoplasma, which is having about a genome size of in the range of 10 raised to 6, 1 into 10 raised to 6. Whereas gram-positive and gram-negative bacteria, the overall genome size may be in the range of 10 raised to 6 to 10 raised to 7. And in the case of fungi, algae, and molds, it is in the range of 10 raised to 7 to 10 raised to 8 orders of magnitude. And crustaceans, echinoderms, insects, mollusks, birds, etc., that may range from 10 raised to 8 to 10 raised to 10. And in case of humans, we can see in the case of mammals, which is represented here. This is the range of mammals here. And that is about, in case of humans, especially we know that it is a 3 billion base pairs. That is the genome content of the humans. So 3 billion base pairs can be represented as a 3 into 10 raised to 9 base pairs. So this is a 3 into 10 raised to 9 base pairs. And in case of amphibians, you can see that about 8 into 10 raised to 8 into 2. This value starts from about 8 or 9 into 10 raised to 8 to about 9 into 10 raised to 10. So that is the range. It's a broad range of genome size is available for amphibians. And for the flowering plants, when we come to the flowering plant, it is having a very broad range, which may range from 10 raised to 7 to 10 raised to 11. So that much size difference is there in the case of the genome size of the flowering plants. So the summary of this slide is that various genomes or the various organisms of the phylum in the system, you can see that there are, for example, all the organisms under various phyla of the kingdoms or animal or plant kingdoms may have different genome sizes, starting from mycoplasma to the flowering plants, which may exist in the higher order of the genome content. So there, within the same phylum or the same group, we can have various ranges. Especially in the case of mammals, we have only a narrow range here, whereas in the case of flowering plants, we have a very broad range, which ranges from 10 raised to 7 to 10 raised to 11 size of a gene. So the summary is that the genome size varies within the same family or the same group of the organisms, irrespective of their genus
there are varying sizes of the genomes, which is question interest. Okay. So you can't say that a plant will have such a specific genome size, or an animal is having such a specific genome size, or an amphibian is having such a specific genome size. It is having all the members within the same phylum may have a very broad range of genome sizes. Some may be very small, some may be very big. Now, the C value might be expected to increase with the biological complexity of the species because of the requirement for more gene counts. So usually we expect that the genome size or the C value, it is expected to increase based on or depending upon the biological complexity. And we assume that, for example, a higher order organism like a human or some other mammal, which may be a very complex organism compared to the primitive organisms or a plant or a single cellular organism, a higher order organism like mammals is having a very complex biological system which demands many more genes for the control and existence of the system. So normally we assume that the number of the genes or required for such higher order organisms or the organisms with higher biological complexity is it may require more genes. So the C value is normally expected to have it needs to be higher in the case of higher order organisms, which is because we are expecting more gene content in such a complex systems. So that's what we normally expect. Broadly, it is true. And the concentration of the DNA in vertebrates is greater than that of the invertebrates, and generally it is greater than that of the fungi and bacteria and viruses. For example, in the case of a virus, it is maybe generally it is having a smaller genome, and in the case of a higher order vertebrate, maybe generally it's having a larger genome. But it is not always true. The minimum genome size within each phylum appears to increase with the proportion of the biological complexity. Usually, we expect that the minimum genome size within the same phylum, it will increase, or we are expecting that it will increase based on the biological complexity, or depending upon the biological complexity. If it is a complex one, we are expecting a larger size of the genome. But there are extraordinary differences in the C value between similar species, generating a spread of the C values within each phylum. That's why in the case of flowering plants, we have 10 raised to 6 to 10 raised to 11. Very smaller genome to very larger genome were present within the same phylum. And even in case of amphibians also, we are having at least 2 orders of magnitude difference in the case of the genome size of the members within the same phylum, that is amphibian. So this is this is something that we are not expecting generally, because we are expecting a higher order of the C value or higher value of the higher genome size or higher C value, depending upon the biological complexity or a proportionate size of the genome with respect to that biological complexity is normally expected, which is not always the case. The largest insect genomes are bigger than the largest mammalian genome. The largest genomes of all beings, actually it belongs to the flowering plants, not vertebrates or animals like humans. So the largest genomes were actually present in flowering plants, and the largest insect genome is also it is actually bigger than that of the mammalian genomes or even humans. That means we can't say that the insect is having a higher biological complexity than that of the humans or a vertebrate. So the summary is that the genome size and the biological complexity is not directly related. The C values vary commonly among the species, and in animals, they range more than 3,300 fold. So that much difference is there in the genome size within the animal kingdom. So in animals itself, if one organism is having a size of X in the genome, and the other higher end organism or higher size genome animal may have 3,300 fold increase in the overall genome content of the concerned higher order organism. And in land plants, they differ by a factor of about 1,000. So the lower range of the land plants and the higher genome size species of the land plants is having about 1,000-fold increase in the genome size, or that difference is there. So the protist genomes have been reported to be very more than 3 lakh fold in size. So that's a lower size genome of the protist, and the higher size genome of the protist differs by 3 lakh fold magnitude. So that much difference is there, but it is undisputed fact, because the higher end range or the largest genome of the protist range is actually attributed to some amoeba. It's actually a questionable one. So still, we say that within the protist itself, there are very small genome size organisms and very large or 3 lakhs fold larger genomes of the protists were also reported. So variation in the C values bears no relationship to the complexity of the organism or the number of the genes contained in its genome. So in conclusion, we can say that the variation in C values bears no relationship to the complexity of the organism or the number of the genes contained in the genome. And it's an observation that was deemed only or fully counterintuitive counterintuitive before the discovery of non-coding DNA, and which became known as the C value paradox. So the C value paradox is actually a fact that the C values bears no relationship to the complexity of the genome or the total number of the genes present in the organism. In higher order organisms like humans or vertebrates, we are expecting more functional genes for operating all these complex systems, but the C value may not be correspondingly high. So the C value has no relationship with the complexity of the organism and the number of the genes contained in its genome. And this is normally against the normal assumption that there is a direct relationship between the content of the gene or the genome size and complexity or biological complexity of the organism. It was normally expected, but it is not the case. And this is known as the paradoxical effect, known as a C value paradox. And now this C value paradox is actually reported earlier, but now we know it is no more a paradox. It's no longer a paradox because we can explain why it is happening. And that is because of the non-coding or gen DNA materials which is present in such genomes. In humans itself, we say that only about 1 or 2% of the entire genome, or the 3 billion bases, corresponds to coding regions. Coding regions, which actually calls for 25,000 to 30,000 genes, which is identified. That's all the entire size of the human genome. So only 2 to 3% is actually represented as functional coding genes. The remaining 98% is actually non-coding regions or repeated regions or various other control elements, which is not actually contributing to a specific gene function. And in other organisms, maybe a higher order organism with very large genome, the percentage of such non-coding regions will be much higher, and that's what causing the this C-value paradox or paradoxical element. It increases the size, not the biological complexity. So we can say that the biological complexity may be directly proportional to the coding regions. If you are saying that, there will not be any paradox. But the overall, if you are taking the overall genomic content and if you are comparing it with the biological complexity, there will not be any direct correlation. That is because of the difference in the percentage of the non-coding regions which is present in the genome. So with the higher value of the non-coding regions in some amphibians or such insects or the flowering plants, and depending upon that high percentage of the non-coding regions, the genome size will increase, but the overall complexity of the phylum will not increase, it will be the same. So this is about the C-value paradox. Now we can explain, so C-value paradox doesn't exist anymore. Still, this term is, it remains in common usage. This C-value paradox is still used, but we can explain, so it is not, it is no more a paradox. In eukaryotes, a repetitive DNA accounts for a much greater proportion of the gene. And this varies between different species. You can see that about 5% of the microbial eukaryotes, some microbial eukaryotes, and it's about 40 to 60% in mammals, 80% in some flowering plants. So this repetitive DNA content varies. Usually, microbes and microbial eukaryotes or single cellular eukaryotic systems like yeast or something, the repetitive DNA content will be much less. But in mammals, it is more than half. And in flowering plants, it is again higher. So that repetitive DNA content is actually some of the main contributing factors to the increase in the genomes. The reassociation experiments originally show that these eukaryotic DNA could be divided into 3 components. And they are unique DNA sequences, moderately repetitive DNA sequences, and highly repetitive DNA sequences. This, the current understanding of the complex genomes reveals several factors that would explain the classical C-value paradox. So it is not only that a single word, repetitive DNA sequences. Now we can attribute many more regions of the DNA that contributes to overall increase in the genome size. And this may be introns in the genes, regulatory elements of the genes, then pseudogenes, multiple copies of the genes, intergenic sequences, then repetitive DNA. So there are various types of such non-coding genes or giant DNAs or such regions of the DNA which is not actually contributing to the biological complexity were present in the eukaryotic system. So these are all some of the genome or the parts of the genome that actually contributes to the actual C-value paradox. So if you can explain that these are the causes for the increase in the genome size, then it is no more a paradox. Okay. So the complexity of a genome is identified as the total amount of unique sequence DNA and may be expressed in physical units. So the size of the genome or the C-value can be expressed in various units like number of base pairs or the quantity in picograms, the weight of the DNA. So in various terms, we can, or using various units, we can explain, or we can quantify, you can express the overall quantity of these genome size or C-value. The difference in C-value within phyla appears predominantly to reflect differences in the repetitive DNA content. So these differences within the phyla appear predominantly to reflect differences in the repetitive DNA content, which does not contribute to the genome complexity. So that's what we are going to summarize, where the C value, the difference in the C value within the same phyla is actually attributed mainly to the repetitive DNA content and other non-coding regions which is present. But within the same phyla, if it is different, usually all of the parameters will be same, but still repetitive DNA content within the same phyla will be much higher. So that difference may be attributed to repetitive DNA content within the same phyla. So it will also cause a difference in the overall C value of the members within the same phyla. Now, when repetitive DNA has been taken into account, however, there appear to be disproportionate differences in the genome size between species of similar biological complexity, and especially when comparing certain groups of unicellular organisms. So there are in the case of these unicellular organisms, there are disproportionate differences in the genome. Some examples were also given there. That is Saccharomyces cerevisiae, and its size is about 13.5 megabase base. Whereas Saccharomyces bombay, a related species, is a 20 megabase base. That difference is almost about 1/3 of the size is higher in the case of Saccharomyces bombay. So within the same species itself, we have a very significant difference between the genomes. And both organisms are similar, is having similar structural or biological complexity and little repetitive DNA. So they don't have any large quantity of the repetitive DNA, and their biological complexity is same. They are having the same, or they are represented within the same genus. Along all these factors are still remaining, but still there is about 1/3 of the difference, overall size difference is there between these related species. The difference is in non-coding unique DNA sequence, that is intergenic DNA segments and introns, and this difference actually it is contributed by the not with the repetitive DNA, but with the intergenic DNA segments and introns. Within the gene itself, there are intron regions which is present. You know that. And with the difference in the size of the intron sequences, the overall length of the DNA or gene will increase, and that will contribute to the difference. And 40% of the Saccharomyces bombay genes contain introns, whereas only 4% genes of Saccharomyces cerevisiae has introns. So that difference within the same gene will cause a difference in the size. So it is not about repetitive DNA or such other region. Even the difference in the quantity of the introns within the same genus may contribute to significant difference in size difference within the same genus. One example is classical example is the Saccharomyces cerevisiae and Saccharomyces bombay and their size difference. Then the next one, there are larger, numerous intergenic regions and introns in higher eukaryotes, leading to increase in the average size of the gene and the distance between the genes. So these intergenic regions and introns in higher eukaryotes, actually this causes larger differences in the average size of the gene. And one more thing is that between the overall distance between the genes within the same genome will also be considerably affected by the presence of these intergenic regions or introns, which will increase overall size, and proportionately the distance between the genes will also increase if there are larger intron sequences in between the genes. So this is actually normally seen in the case of the eukaryotes. So this is about the distribution and function of DNA sequence count. In bacteria, most of the genome is unique sequence of the DNA. There is no more such repetitive DNA or junk DNA or such non-coding regions. Normally, it is not present. Mostly the bacterial genome is very tightly packed, so most of the regions were useful, and we can't expect much unwanted regions or non-coding regions or repetitive DNA or such regions may not be present in the case of bacteria. So in bacteria, most of the genome is a unique sequence DNA representing genes and regulatory elements, not unwanted ones. And some genes are and other sequences are repetitions, but the copy number is generally low. And if there is some repetition in the bacterial genome, that will be very less. That means the copy number of the repetitions of the bacterial genes will be much less. Examples of such repetitious sequences include the rRNA genes. One example. And 7 such genes of rRNA genes were present in E. coli. And transposable elements such as IS elements and occasionally sequence motifs may be moderately repetitive. So these certain sequences were moderately repetitive sequences that were maybe also present in prokaryotes. But the previous example that we mentioned, for example, that rRNA gene in E. coli, there are 7 such repetitions, but it is required because rRNA is used in case of ribosome. Ribosomes were always required in sufficient quantities, or it's a structure as a structural component of rRNA or ribosome, that they were actually required in sufficient quantities within the cell always. So for that, a single copy of the gene may not be sufficient for producing sufficient quantity of rRNA. And that's why such genes in prokaryotes, even though it is present in larger numbers, actually it is required. This is based on the overall demand of the rRNA copies that is required in the cell. The number of genes were of a larger number of copies, like 7 repetitions were present for rRNA genes. And the general concept is that in prokaryotes, we can't expect such junk DNA or the DNA regions that is normally unnecessarily expanding the size of the genome. So this paradoxical effect normally we expect only in case of where eukaryotic systems, not in prokaryotes. And the E. coli genome contains many copies of 2 repetitive elements, that is ERIC. ERIC means enterobacterial repeated internal consensus, and another one, a rep sequence, that is a repeated extragenic palindrome. So these 2 are repetitive sequences that is present in case of E. coli. So we can't say that repetitive DNA is not present in prokaryotes. There are, but only very few. Compared to the eukaryotic system, the repetitive DNA sequences in eukaryotic systems were very less, like some of the functional genes like rRNA genes, or in case of E. coli, for example, ERIC sequences and rep sequences, which is these are used for the phylogenetic analysis of E. coli and their studies. This ERIC and rep basic PCR for identification and of the E. coli species is actually present. So PCR based amplification of ERIC sequences, just like the fingerprints. Usually, we know that DNA fingerprinting is based on repetitive sequences. Each and every individual will have their own fingerprint on locations, based on which the DNA fingerprinting technique develops. And similarly, for E. coli, if you want to analyze or differentiate into E. coli species, their phylogenetic technique, if you want to compare, then ERIC or rep sequences can be used so that there will be differences in the number of repetitions within the different species of E. coli. So this is these are two well-known repetitive sequences in E. coli genome. They are eric sequences, that's intrabacterial, repeated internal consensus, and a rep sequence, that is a repeated extragenic parameters. These two are present in E. coli. And in the 1.8 megabase per hemophilus influenza genome, there are about 1,500 copies of 30 base pair DNA uptake site. So that's another example of a repetitive DNA sequence in case of a prokaryote. In case of hemophilus influenza, we have about 1,500 copies of 30 base pair DNA uptake site. That DNA uptake site, which is about 30 base pair line, is repeated for 1,500 times. Together, however, repetitive DNA accounts for 4% of bacterial genomes, and the genome size is a direct reflection of the complexity. So that means in prokaryotes, only about 4% is repeated. So it is comparatively much less. So it won't cause any paradoxical effect because the genome size is almost proportional to the complexity of the organism. Whereas in case of other hybrid organisms, the genome complexity doesn't have any relationship with the size of the genome because of such repeated sequences and the sequences that is mentioned of the step previously. So these are the facts. So this is about the C-value paradox. Any doubts?